Welcome to Evolutionary Biology. Today, we are diving into two of the most fundamental forces that drive evolution: migration and mutation. If you have ever wondered how new traits appear out of nowhere, or why certain populations look so similar despite living far apart, you are in the right place. Let us begin our journey. To understand evolution, we must first understand where variation comes from. Imagine you are copying a massive, thousands of pages long family recipe book by hand. No matter how careful you are, you will eventually make a typo. In biology, this recipe book is the DNA, and the typos are called mutations. When cells divide, random errors inevitably occur. These errors create entirely new versions of genes, introducing brand new alleles into a population's gene pool. This is the fundamental starting point of all evolutionary change, acting as the spark that ignites the entire process. Now that we know what mutations are, we must ask, why do they matter so much? Without mutation, evolution would completely grind to a halt. Natural selection needs variation to work. It needs different traits to select from in order to adapt to changing environments. If every individual in a population were genetically identical, no trait could be favored over another. The population could never evolve to survive new diseases or climate shifts. Therefore, mutation provides the essential raw material upon which all other evolutionary forces act. Since mutation is the engine of variation, let us look at how we can measure its impact mathematically. The formula on screen calculates the change in allele frequency over a single generation. Delta P represents this net change. It depends on two competing rates. The variable U is the forward mutation rate, meaning the probability that allele capital A mutates into allele lowercase a. The variable V is the backward mutation rate, where lowercase a mutates back into capital A. The variables P and Q represent their current frequencies. Physically, if the forward mutation rate U increases, the frequency of allele capital A will decrease more rapidly as it is lost to mutation. Let us see this formula in action with a concrete example. Suppose a population of beetles has an allele capital A with an initial frequency of 0.80 and an allele lowercase a at 0.20. The forward mutation rate is 0.0001. The backward mutation rate is so small, we assume it is 0. We want to find the change in frequency of capital A. Because the backward rate is 0, we drop that term. We simply multiply the negative forward mutation rate by the frequency of capital A. The result is negative 0.00008. This tiny number shows that mutation alone is a very slow evolutionary force. While mutation creates new alleles slowly, another force moves them around rapidly. Imagine two isolated islands, one where everyone wears red shirts and another with blue shirts. If people from the blue shirt island move to the red shirt island, they introduce blue shirts to a place that never had them. In evolutionary biology, this movement of individuals and their genetic material is called migration, or gene flow. Unlike mutation, which creates alleles from scratch, migration simply redistributes existing alleles across different populations. It acts as a powerful homogenizing force, mixing the genetic paint between geographic areas. This mixing process brings us to why migration is incredibly important. It prevents populations from diverging too much. If two populations of the same species are completely isolated, they will eventually accumulate different mutations and adapt to different environments, potentially splitting into two separate species. Migration acts as a glue that holds a species together by constantly sharing genetic updates. Additionally, migration can rescue a struggling population by introducing beneficial alleles that evolved elsewhere, increasing the overall fitness of the receiving group. To model this genetic mixing mathematically, evolutionary biologists use the one-island model. This formula calculates delta P, which is the change in the allele frequency on the island after one generation of migration. The variable m represents the migration rate, which is the proportion of the island population made up of new migrants. The variable p sub m is the frequency of the allele in the mainland population, and p is the initial frequency on the island. The formula tells us that the change is directly proportional to both the migration rate and the genetic difference between the populations. A larger genetic gap means a faster change. Let us apply the one-island model to a population of mice. We are given a mainland black fur allele frequency of 0.70 and an island frequency of 0.20. The new migrants make up 10% of the island, so our migration rate m is 0.10. We want to find the change in the island's allele frequency. First, we subtract the island frequency from the mainland frequency to find the genetic gap, which is 0.50. Multiplying this by the migration rate gives us 0.05. This single generation of migration causes a massive shift, making the island look much more like the mainland. Now that we have calculated the change, let us visualize exactly what is happening in the one-island model. On the left, you see the large mainland population, represented by a massive pool of mostly blue dots, indicating a high allele frequency of 0.70. The thick arrow represents the migration rate, m, showing the one-way flow of individuals. On the right is the smaller island population, initially dominated by red dots with a low blue allele frequency of 0.20. You can clearly see how the influx of mainland migrants will rapidly shift the genetic makeup of the smaller island. We have seen how variation is created and shared, but this variation comes with a cost. Imagine a factory that produces highly efficient cars, but occasionally a machine glitches and produces a defective engine. Because mutations constantly introduce slightly defective or deleterious alleles, no biological population is ever perfectly adapted. This constant burden of less than perfect genes is what biologists call genetic load. It is a measure of the cost a population pays for maintaining genetic variation. Most new mutations are harmful, and they lower the overall average fitness of the population. This constant influx of defective genes leads to a fascinating dynamic known as mutation selection balance. While natural selection works hard to weed out harmful mutations, the mutation process constantly introduces them back in. This tug of war creates a steady state. Understanding this balance is crucial because it explains why genetic diseases persist in human populations. Conditions like cystic fibrosis are caused by deleterious alleles. Natural selection has not eliminated them because new mutations constantly replenish these disease alleles in the gene pool. It highlights the fundamental limits of natural selection. To understand the exact toll these mutations take, we can quantify genetic load mathematically. The formula on screen calculates L, the genetic load, which is a dimensionless number between 0 and 1. The variable W max represents the fitness of the optimal, most successful genotype in the population. The variable W bar represents the mean, or average fitness of the entire population. We calculate the difference between the maximum possible fitness and the actual average fitness, and then divide by the maximum fitness. If the average fitness decreases due to an influx of bad mutations, the genetic load will increase, indicating a less healthy population. Let us calculate this evolutionary tax for a population of plants. We are told the best possible genotype produces an average of 100 seeds per plant, so W max is 100. However, due to deleterious mutations, the average plant only produces 85 seeds, making W bar 85. We want to find the genetic load. We subtract 85 from 100 to find an absolute loss of 15 seeds per plant. Dividing this by the maximum of 100 gives us 0.15. This means the population is operating at a 15% deficit in reproductive output due to the constant process of genetic mutation. We have covered a lot of ground today, so let us review our key takeaways. First, mutation is the ultimate creator. It generates brand new alleles through random errors in DNA replication, providing the raw material for evolution. Second, migration is the great mixer. It rapidly moves existing alleles between populations, preventing them from diverging into separate species. Finally, we learned about genetic load, which reminds us that evolution is never perfect. The constant introduction of mutations creates an unavoidable cost. You now have a solid understanding of how migration and mutation shape the incredible diversity of life.